Hello everyone and welcome to ITLS Academy Empower the Youth. So today in this lecture we will going to study about a new experiment, that is to check the presence of antibiotics in milk sample through DSM DELVO test. So in this particular lecture we will going to study about how you detect the presence of antibiotic in different dairy product. That is, today's study is based on milk. So how you detect the presence of antibiotic in milk sample. So let's see the introduction part and let's see what is it. So antibiotics have been used for the prevention and treatment of common diseases and for prophylactic purpose in dairy animals. However, in recent decades, it can become a matter of concern due to the widespread belief that there has been abuse or misuse of these drugs in animals, and that is misuse has led to the presence of residue in derived food such as milk and dairy product. So antibiotics, it is being used in different types of dairy product, especially for the prevention as well as for the treatment of various type of diseases, especially a prophylactic purpose in dairy animals. But the recent decade, due to the alarming you can say, people are becoming more conscious, and now different types of studies are being there to detect whether a sample is pure or not, means whether a food that we are consuming is pure or not, whether the antibiotic it is using in that particular sample is in appropriate manner or not, right? So in the recent studies it has been shown that the use of antibiotic it becoming a matter of concern. How? Because these antibiotics is used in animal, and it is being used as well as it is used in a misway, means it is being misused in these drug in animals, and this misuse lead to the presence of residues that is derived from the food, like such as milk and dairy product. Means in simple word, if I would like to explain this, that this antibiotic nowadays is people are using antibiotic as a misused, means they are misusing this antibiotic and in the dairy and milk products. So antibiotics are defined as a naturally occurring and synthetic compound with antimicrobial activities that have been applied apparently orally or topically. So what is now antibiotics? Why it is being used? So antibiotic, it is nothing but it is naturally occurring, and sometimes it is semi-synthetic or synthetic compound that have a antimicrobial activity, means they have a antimicrobial activity, means they act against microorganism, that is basically they are used to kill different types of microorganism or to eliminate different types of microorganism. So it is used for the with the it has a antimicrobial activity, and they have applied parentally, orally or topically. So antibiotic have been used in livestock care for more than 60 years for the prevention and therapy of common pathogens like and the prophylactic lactic purposes. So antibiotic is being used since 60 years. Now it has been 60 years that people are using antibiotics in dairy and milk product, and it is being used to prevent and do the therapy of different types of pathogens, and like respiratory diseases, like mastitis diseases, like foot diseases, etc. So it is basically used to treat these diseases, and also prophylactic purposes in dairy and milk products. So it is being used, but now because of you can say the huge population, to meet the demand of the huge population, some what it is being, it is now misusing, Now antibiotics is misused in milk and dairy products. So our aim is to check whether our food sample have antibiotics or not. So it is a raw diagram just to let you understand what does it means. So antibiotics are used in dairy farms, where antibiotics uses to prevent and treat infection can accumulate in the manufacturing and select for the antibiotic resistance bacteria which can be transferred. So antibiotic, if you see in this diagram, it is used in dairy farms. Why it is used? Just to prevent and treat various type of infections, right? So what it does do, so it have contaminated meat and dairy products, right? So microorganism, how, why it is used? Because microorganism is there that can contaminate the meat and dairy products. So because of that, antibiotic is used. So it is used to other farms, animals and lives. It is how it is used via farms, workers and food processor, and to the land and water resources via applied antibiotics, biosolids, manufacturer runoff. So it, in this way it is being used. Now today's aim is to check the presence of antibiotics in milk sample through DSM DELVO test. So what is DSM and DELVO test? So if we talk about DSM or DELVO test, so it is basically a test that is used to check the presence of antibiotics. Okay, and this type of DELVO test, it give a accurate result in under 3 hours. Means under 3 hours they can give you a accurate result. So why it is used, means what is the purpose of using this test? So we will going to study this in detail. So before that, let us understand what is DELVO test. So DELVO test are the standards test for the determination of the residual amount of antibiotics, or you can say sulfa substances in the milk. So I'm just writing what is DELVO test. You can note it down somewhere. So what is DELVO test? DELVO test. So what is DELVO test? They are standard test for the determination since the space is little less, so I'm just removing this. So you can note it down. DELVO test. What is DELVO test? So they are the standard test for the determination. Further I am writing determination of residual amount I am writing in short, amount of antibiotics and of course the sulfa substances in milk. Okay, so this is the definition of DELVO test. Means if in examination or in interviews, if they can ask you what is DELVO test, so DELVO test is nothing but it is a type of a test that is used to detect or the presence of antibiotics and sulfa substances in the milk sample or dairy samples. So now let's move further. So the theory says antibiotics contamination in milk can seriously affect consumers' health and cause allergic reaction to residue or by development of resistant strain of microorganisms. So since antibiotics have an antimicrobial activity properties, but if it is used in a misway, or it is using in an inappropriate manner, so they can create a serious health effect, or they can cause an allergic reaction in consumers, and therefore subsequently antibiotic contamination in milk can also cause significant economic loss for the producer and manufacturer of milk and milk product. Okay, apart from causing a damage to the human beings, means causing a damage to the consumer, they can also cause a significant economic losses for the producers as well as the manufacturer of that particular milk and milk product industry. So for the determination of antibiotic residue in milk, use commercially available antibiotics. So there are different types of detection kit are there. Those type of kit is used to determine whether the antibiotic residue is present in the milk sample or dairy sample or not. So what are the different types of kits that are available in the market to check? So the first is DELVO Express BL second is that kit name. Then they have Copen test, they have Charm Farm test or Charm Aim 96 Beta Star kit. So there are different types of kit that are available in the market, so you can purchase them, the detection team purchase this kit, and according to by the help of this kit they can detect or determine and whether the antibiotic residue is there in milk sample or not. So the growth of bacillus stereothermophiles spore at 64 degrees centigrade initiate an identification process which cause the turning of a pH indicator from yellow purple to yellow. So the presence of antimicrobial substances will cause delay of inhibition of the spore depending upon the concentration of residue. So in the presence of residue spore, they will not multiply and pH indicator will remain purple. So we will check how this will happen. So these are certain different types of charm farm detection kit. The first figure is of the charm farm test kit. This kit look like this, and it is basically used to check the presence of antibiotics in the milk. Second is DELVO Express kit. So a DELVO Express kit look like this, and through the help of this kit you can detect the presence of antibiotic in different milk sample. Now let us understand how DELVO SP microbial test is. Means what is DELVO test SP microbial test? So milk sample were analyzed during 6-hour period after collection. Okay, firstly you take a milk sample and you analyze that milk sample for 6 hours period by DELVO test SP DSM food specialities milk dairy ingredient delft, and means it is a kit of the Netherlands. I am just taking an example. So the method was carried out during according to the instruction of the manufacturer, means the manufacturer give instructions. So according to the instruction, the method was carried out. Thus you take 100 microbial milk sample, and this milk sample were added to the individual DELVO test cup ready prepared, containing beta stereothermophilus and calidolactis and an indicator in solid medium, one nutrient tablet containing carbon and nitrogen source facilitating the growth and metabolism of the test organism was added to each cup. After you add that, the plate were sealed, means you sealed the plate with the tape that has been supplied. Then the plates were incubated in the water bath for 64 at the 64 degrees centigrade plus minus, and for 3 hours, and the manufacturer instruction they record the values, or they observe the sample. Now for this particular test, Now we will going to see what all materials is being required. So the first material that is required is DSM DELVO test kit. The kit look like this, as you can see in the image. Then you need a raw milk sample, then you need a water bath, then you need a agar well strip. The procedure says you first add one nutrient tablet to each of the agar well in the strip. Means, as you can see this agar well, so you add one nutrient tablet to each agar well in the strip. Okay, after adding the one nutrient tablets, what you do, you inoculate 100 ml of milk, the raw milk, into the agar well plus the nutrient tablet that is present. Then after inoculating the 100 ml of milk into the agar well, and already there is a nutrient tablets, so what you do, you seal the well and you set it for the incubation. Then after sealing and setting for the incubation, you incubate the strip or well in the water bath. What should be the temperature? 64 plus minus 0.5 degrees centigrade for 2 hours 30 minutes. At the time the negative control has been changed to yellow. Okay, now we will see what is the result. So what you have to do, you have to incubate the well in the water bath at 64 degrees centigrade plus minus 0.5 degrees centigrade for how many hours? 2 hours 30 minutes. Then you have to examine the strip or well for the color change from purple to yellow. Now you, after incubation, after 2 hours means 2.5 घंटे के बाद, you check or you examine the color of the well, whether the color is changed from purple to yellow or not. So a yellow reading indicate no inhibitory substance are present. A purple reading indicates that the antibiotic residues are present. Okay, so if the well have a purple color, so it indicate yes, the antibiotic residue is present. But if it is yellow, then it indicates that there is no antibiotic substances or residue is there. So yellow, if the color is yellow, indicate there is no antibiotics residue, there is no antibiotic residue. And if it is purple, then yes, antibiotic residue is present. So you should always know. So here 2 or 3 things need to be understand, that at which temperature it is incubate, 64 degrees plus minus 0.5 degrees centigrade, and how much time it is needed to incubate, 2 hours 30 minutes. Okay, so for the best sensitivity, a control time reading is advised, usually using a negative control sample. So for the best sensitivity, a control time reading is advised, and a sample incubation period is crucial to the accuracy of DELVO SP test method. So this particular applies the detection of sulfamides as a sensitivity of to the sulfamides is greatly reduced by increased incubation time. So an incubation time for the 2 hours 30 minutes is recommended, with 15 minutes extension time for the test in case of suspect sample. So it is recommended that you extend the incubation time from 2 hours 30 minutes to again 15 minutes more. For reading time, that is 2 hours 45 minutes or 3 hours, the sensitivity test will be diminished. The use of antibiotic free skim milk powder as a control is advised. The essential that the correct temperature is maintained in the water bath, the use of proper water bath lid, the slope lid is advised, the temperature control in the water bath. So so in this particular paragraph it is being telling that you should be taking, because it is a very sensitive test, so you should always have to be very conscious while performing this experiment in your respective laboratories for the correct and correct reading, or not reading, correct result. Why? Because if there is ऊपर नीचे हो जाता है कभी कभी, अगर temperature is not properly maintained, if water bath temperature is not properly maintained, if the wells are not properly sealed, so there are chances of a inappropriate result. Okay, so for the best result, what should be what precautions a person should take, means the well should be properly, firstly it should be properly sterilized. Secondly, it should be properly sealed after you pour the milk and the nutrient tablet. Then the temperature should be properly controlled, means there should be a proper temperature control. The water bath temperature should be correct. Okay, and there should be the milk or the sample should be incubated for a perfect temperature, for perfect time, that is 2 hours 30 minutes, and you should always extend it for 15 minutes for the better accuracy. So these are certain precautions that you should always take while performing this experiment. So further you should make a observation table like this. Here you put your sample name and the color. So as you can see the result and interpretation, if so this type of thing that you will get as a result. So if you can see the first image, that is yellow, so yellow shows that no antibiotic is present. Then the second image we will talk later, then you see positive one, means a purple, that shows antibiotic is present. But if your result is showing like this, means there is a purple also, there is a negative also, oh sorry, there is a yellow also. So this type of result shows invalid result, मतलब कुछ नहीं है, it involves a बिल्कुल बीच का ही result. So you should not consider this result, and it is always recommended that yes, you should always मतलब see for the negative and positive result, that is yellow and purple. Okay, so this is how you detect the presence of antibiotic in different types of milk and dairy products. So apart from this, DELVO test kit is also used in treating other liquid products also. Milk is one of the product. Apart from milk, you can also check for the yogurt, you can also check for different types of chocolate milk also. You should they are also used where you check the urine. They are also used in detection of raw meat juice, blood serum. So these can also be tested through these type or through with this type of kit. So that brings to the end of the lecture. I hope you have understood the lecture. So let's quickly understand again what all we have covered so far. So as we have covered the antibiotics, so you must be aware of antibiotics, the definition of antibiotic. Sometimes in examination or interview they can ask you what is antibiotic or what do you understand by antibiotics. So you should always be aware of these questions. And regarding the test, of course they can ask you name any 2 or 3 detection kit that are commercially available and they are disposable one, and which type of bacteria can grow, and how this test take place, means what is the procedure of this test, how to detect the presence of antibiotics, right? So you must be aware of these procedure, and they can ask you about the what do you understand by DELVO test, SP matlab microbial test, so you must be aware of this. And they can ask you what is the result, means if there is a yellow color, they can ask you like this: if there is a yellow color after the procedure that has been happened, so what a yellow color indicates? So they indicate there is no antibiotic. And if it is purple, so it indicates the antibiotic. And if there is the mixture of both color, so that test is invalid. So you must be aware of these questions. So these questions must be asked, should matlab they can ask you in the examination, so you must be properly prepared for these type of questions. And yes, so that brings to the end of the lecture. If there is any doubt, anything related to the lecture, or um, you want to ask anything, so you can simply leave your queries in the comment section below. In the next lecture, I'll be answering the queries. And if you have any doubt related to any of the courses, students, if you want to enroll in any of the courses, so you can simply call on our helpline number, that is 7080833450. For better understanding, you can also visit our website, that is www.itlsacademy.com. You can follow ITLS Academy on Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and you can get our this app from Google Play Store. And yes, student, don't forget to press the bell icon to get more update, and please subscribe ITLS Academy, because on this channel we upload different types of lectures on different topics. So you can enroll you can press the bell icon. So thank you for watching. Please do like, share and subscribe, and don't forget to press the bell icon to get more update. Thank you.